All right, this is the second part of your proteins notes that I would call enzymes. So let's back up a little bit. There were two types of proteins, fibrous proteins, and we agreed that those were really strong. They were insoluble in water, so they do not dissolve in water. And there were the examples there. And then globular proteins was the other type. And these are big globs. And we mentioned that enzymes are globular proteins because they have very complex shapes. So this is where we're going to start with enzymes. For enzymes, most enzymes are proteins. They increase the speed of chemical reactions in the body. They digest food, and they control reactions in the cell. They are very specific. Each enzyme has a specific substrate that it may act on. So it's kind of like a lock and key, where there's one key that opens a specific lock, is the enzyme-substrate relationship. The substrate is just the molecule on which the enzyme can act. So enzymes are affected by temperature and pH. They're only active, they only work at specific temperatures and pHs. So remember, their jobs are to increase the speed of chemical reactions, digest food, control other reactions in the cell, and they only work at very specific temperatures and pHs. They're very picky. So this is an example that I'm going to show you, kind of a model of an enzyme structure. Enzymes are globular proteins, like I said just a moment ago. The way they fold up creates a place called an active site. This is kind of like the particular place in a lock that a key actually goes. The nature and arrangement of the amino acids in the active site make it very specific. So here is an enzyme called catalase. One thing you need to write down in your notes that there isn't a place for is that most enzymes are called ACEs. And they may act on their particular substance. So catalase is this huge enzyme, and its substrate is H2O2, which is actually hydrogen peroxide. And so the active site is this particular spot right here where H2O2 will bond or bind, and it fits right in there just like this model substrate fits into this active site. So they work very particularly together, the enzyme and the substrate. And remember, enzymes are usually ACEs. So let's look at an example. The example is lactase as the enzyme. It acts on lactose, which is a carbohydrate, and it would be a disaccharide, right? Because it's made of glucose and galactose together. So this is lactose, and lactase is the enzyme that breaks lactose down. So one more time, lactose is a sugar. It is milk sugar, but it is a carbohydrate, and it is a disaccharide specifically. And your body cannot use this disaccharide lactose. It would like it to break it down into galactose and glucose, but it can't do so on its own. And so this reaction would be called not dehydration synthesis, but hydrolysis, because we're going to break this down. I almost said dehydration synthesis. But it's called hydrolysis because we want to break it down, but it won't break it down on its own. It needs a helper. And so when we want to break this lactose down, we use an enzyme called lactase. So if this doesn't happen in the body, because you don't produce any lactase, then the lactose will not get broken down into galactose and glucose. It will turn into lactic acid, which causes pretty severe cramping in your abdomen, in your intestines, and it can cause gas and diarrhea and an awful, unpleasant, uncomfortable, and that's what we call being lactose intolerant. If you don't make lactase, you are lactose intolerant. So, name that reaction. I already spoiled that one. The reaction that breaks down lactose into galactose and glucose is hydrolysis. I keep trying to say dehydration synthesis, but that would be the opposite, right? So hydrolysis breaks it down. This piece is about carbonic acid, and it kind of talks about specifically as the buffer acts in the blood, but I don't necessarily need to talk about that. Excuse me. Nucleic acids is the last. They are the last organic compound. So let's review. We have carbohydrates. Those were monosaccharides, disaccharides, polysaccharides. They all were made of glucose and other monosaccharides like glucose. And they were all made of carbon, hydrogen, and oxygen. Then we have lipids, made of also carbon, hydrogen, and oxygen, but yet in a different type of way. Lipids were larger, and they were amphipathic, polar and nonpolar. Examples of lipids were phospholipids, triglycerides, steroids, pigments, and waxes. Then we have proteins, and proteins we talked about were made of amino acids in chains. We had primary, secondary, tertiary, and quaternary structures of proteins, and we had two main types of proteins, which were globular and fibrous. Fibrous were long and tough, and globular were big globs that make up enzymes. Then I started this presentation, which was about enzymes being proteins, and then how enzymes act on substrates. So, the fourth and final type of organic compound is nucleic acids. And this is the one I told you I'm going to spend very little time on because we spend very many units on it later. So nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And examples are DNA and RNA. Nucleotides make up the DNA that, like this would be a big molecule of DNA, and the actual nucleotides are the subunits of DNA. So if you were to break this huge molecule apart, you'd be breaking it into a nucleotide that looks like this. So you don't have to know anything but that nucleic acids, examples are DNA and RNA, and that nucleotides are subunits of DNA. They make up DNA. And then the rest will be to be continued as far as DNA and RNA go. And that is it.